Yeah, good morning. Good morning, everyone. Karishma, Shantani, welcome to the class. Can you hear me? All okay? Good morning, Manju. Good morning, Manju. Okay. Happy Independence Day to all of you. Okay. So I think we can wait for a few minutes, then we will start. Yeah, thank you so much. Thank you. Okay. So can you hear me? Is it okay? Everyone, can you hear me? Okay. Fine. Okay. So I think we can start. Right? Okay. So one more session, and that session is on ICMR JRF preparation, obviously, and just like we did last day, this session is all about a Q&A on virology. Okay. As I told you that on YouTube I will discuss some model questions that you can have, that you can face in ICMR. Okay. So that's what it is. So Q&A discussion on virology. So I think it's time to start the session. So before going into all those things, let me introduce myself. My name is Shuman Panda, Senior Research Fellow at Department of Biophysics, Bose Institute, Founder Director of Golden Helix Center of Excellence. I qualified CSI Net with All India rank of JRF 73 and qualified these exams as well. And I completed my graduation and post-graduation from University of Calcutta with merit rank of 5th and 3rd, and I have more than 5 years of experience in CSI Net, GET, DBT, ICMR, and all these entrants, and guided more than 200 students in PhD entrances. Okay, fine. Is it okay, Karishma? I think so. So that's my profile. I think all of you know that. Okay. So I think all of you know that I already have taken so many classes on Unacademy, and you will get more than 130 special classes which are there on my profile. So all you have to do, just search my name on Unacademy app, and you will get all those classes, and you can watch all those classes for free. Okay, you can watch all those classes for free out there. Okay, just you have to apply my code Shumon P Live or Shumon P. You'll be able to watch all those classes for free. Okay, and I already completed so many free courses. Okay, only 3 of them I showed here. Okay, so many free courses you will get. Okay, and all those courses are based on PYQ, then theoretical sessions out there. Okay, so it will be very helpful for all of you. Okay, and let me tell you, 11 plus courses 11 plus courses are already completed till that. Okay, 11 plus courses, and you will get detailed theoretical sessions on unit 1, unit 2, okay, unit 3, unit 4, unit 8, unit 12, unit 13. Exactly, these are the units I already completed, and out of all those units you will have 3 units, 3 plus courses, 3 plus courses dedicated to techniques only. And those 3 courses are, let's get ready. This one you will get so many different types of techniques from this particular course, and this one, methods in biology and methods in biology and chemistry. These 3 courses are mainly based on all those techniques and technology you have. Okay, yeah, very good morning Vaishnavi, all of you, welcome to the class, everybody. Okay, and right now I'm dealing with this particular class course where I'm discussing the basic concept of physical chemistry, like atomic structure, chemical bonding, equilibria, reaction kinetics, those regions I'm actually discussing. Okay, just like today also I have another class on bioenergetics part 2. Okay, so if you really want to get all those classes, all my class courses, if you really want to get all those things, all you have to do, you have to subscribe. Okay, and you know, while subscribing you always use this code Shumon P Live. Okay, because once you use this code you will get 10% instant discount from all those fee structures. Okay, and you know why we should go, I mean why you should go for subscription. Okay, because you will have full length courses, MCQ, PYQs, new batch courses every week, dedicated doubt clearing sessions, weekly quizzes in every courses. Okay, and full mock test on all Sundays. Exactly, so that's why you should go for subscription. Okay, and these are the subscription packages out there, you can check, and you can only able these fee structure only when you will apply this code. Okay, remember this one, all the fee structures out there, whether it is plus or iconic, okay, you will only get it once you will use this code, that is Shumon P Live. Okay, don't forget to use this code. Is it okay now? Can you hear me properly? Karishma, Shantani, everyone, is it okay? Is it okay? Can you hear me? All okay? So what is the problem? What's the problem? So is it okay now? Can we? Okay. Okay. So I think we can go for the next question. Okay, next question. So viral envelopes can best be analyzed with which of the following techniques? Go for it. Next question. Viral envelopes can best be analyzed with which of the following techniques? Okay, go for it. Drop your answers. Good morning, all of you, and happy Independence Day to everyone. Okay. So viral envelopes. Okay, fine. So how you can study or analyze viral envelope? Okay, drop your answers, everyone. We discussed almost the same type of questions in the last session as well. Okay. And in that session, I discussed something about these techniques, few of them. Okay, virus envelope. Okay. Just go to the question once more and ask yourself. Viral envelope. Envelope is a surface structure, right? Envelope, capsid, these are all surface structures, right? These are not any internal structures of viruses, right? Okay, so transmission electron microscopy will not be the answer, because transmission microscopy, electron microscopy is for getting the detailing out there from the internal structure. Exactly. If you really want to analyze what is there inside the cell, then you have transmission electron microscopy. Okay, but here what you have to do, so transmission electron microscopy will not be the right answer. It's not possible, because even though if there was a surface electron scanning electron microscopy or used as surface used for surface structures analyzing, or maybe atomic force microscopy, then definitely I will come for that particular option. But transmission electron microscopy will not going to help you here. Okay, so it won't be A. No, it won't be A. Okay, the right answer will be B, because that we can do. You can raise specific antibody against specific proteins of the viral envelopes. Okay, specific proteins of the viral envelopes that you don't get in host membrane. So if even there is a fusion, okay, even there is a fusion between virus envelope and host cell membrane, and if you target a particular virus envelope protein with an antibody, what will happen? That antibody will definitely going to bind with the virus envelope protein only. Okay, so even when the fusion is taking place, you can only visualize the viral protein, because that viral protein will be visualized by the help of the antibody, and that antibody is specifically designed and targeted to the viral envelope protein. That's all it is. Because C and D are nothing it can be correlated with this one. Okay, C. So definitely we can't go for C or D. Okay. So the host range of a virus is determined by what? It's very simple. You can only determine that by the help of the protein on its surface and that of the host. Exactly. Okay, that's the right answer. The proteins on its surface and that of the host. Okay, because the host range is dependent on the receptor specificity, right? There should be some molecules out there on virus, some receptors out there in the host. Okay, so if they can bind with themselves with higher affinity, okay, that will define their host range. Definitely, so it's based on the receptor specificity, receptor ligand interaction. Okay, so there should be one protein on the surface of the virus and one protein on the surface of the host as well. Okay, so that's what it is. That's why the right answer is option A. Okay, fine. Next question. Okay. So in many ways, the regulation of the genes of a particular group of a virus will be similar to the regulations of the host gene. So, which of the following would be will you expect of the genes of the bacteriophage? Go for it. Okay, so answer of this question will be what? They're talking about regulation. Okay, the regulation of the genes of a particular group of virus will be similar to the regulation of host genes. Exactly. Okay, so what you can expect from the genes of bacteriophages? Okay, that's the question. So what you can expect? Definitely control of more than one gene in one operon. Exactly. You know, if you have gone through my operon classes, I told all those things. I discussed. I told that in operon system there can be multiple genes under the same promoter under the same regulator. I mean, that's the difference between prokaryotic gene regulation and eukaryotic gene regulation, right? So I think you all know that. Okay, and is this the same for virus, even for bacteriophage here, or even for bacteria, all the other hosts out there? Okay, mainly prokaryotes, right? So what is the right answer here? That the control of more than one gene, definitely control of more than one gene in our. Okay, so why do RNA viruses appear to have higher rates of mutation? Okay, go for it, everyone. Everyone, drop your answers. Okay, RNA viruses have a very higher rate of mutation. What's the reason behind it? Good morning, Divyankar. And absolutely, you are right. I think you can answer this question from the concept of central dogma, right? That the regulations of the genome does not involve the proofreading steps like DNA replication. Exactly. Exactly. So option B is the right answer, undoubtedly. Because in RNA transcription or the replications of RNA genome, okay, there is no such proofreading activities out there. Option B, absolutely. Option B is the right answer. Divyankar, you are right. Okay, so B is the right answer, undoubtedly. So that's the reason that why you don't expect, okay, that error in DNA viruses, but in RNA virus you can have higher rates of mutation. That's what it is. Okay, fine. Okay, so if you would like all those sessions, and if you really want to have more sessions like this, all you have to do is subscribe Unacademy CSR Education Live YouTube channel. Okay, and don't forget to share all those videos. Okay, and hit the bell icon so that you can get all the notification. Right? Okay. Next question. So antiviral drugs have become useful for usually associated with which of the following properties? Antiviral drugs that have become useful for usually associated with which of the following properties? Everyone, drop your answer. Answer the question. It's very simple. Go for it. Samiksha, good morning. Drop your answers, everyone. Okay, so what's the main target of all those antiviral drugs? I mean, the mode of action I'm talking about. What is the mode of action of all those antiviral drugs? Absolutely, Sijo, you are right. Definitely, you want to block. You want to block. I mean, there is no confusion, you know. You just want to block the reproduction. That's all. Okay. So many antiviral drugs are there. Not so many like antibiotics, but all the antiviral drugs out there in the market right now, they're all targeted to block. One of the steps of central dogma, I mean replication or transcription or translation, whatever. So those are the steps of reproduction, right? Main steps of reproduction. So your antiviral drug should interfere with the viral reproduction. Then only you can get rid of that infection. Okay, obviously. Sijo, you are right. Absolutely. Okay. Next question. Okay. So what are prions? What are prions? Next question. It's very simple one. Go for it. What are prions? Drop your answers quickly. What are prions? You know, I already discussed all those things in my class courses already. Okay, and if you really want to get all those courses, all you have to do is just subscribe, and that's the code. Because if you use this code Shumon P Live, you will get 10% instant discount. Okay. Absolutely. Divyankar, Anisha, all of you are right. Yeah. It's misfolded version of normal brain protein. I mean, basically those trans proteins are being identified in neurons, okay? But the fact is, trans are your own body proteins, but they are misfolded, abnormally folded protein structure. That's all, and that is infectious. Absolutely, all of you are right. Okay, next question. Okay. Go for it. Which of the following represents a difference between virus and virions? Okay, go for it. Okay, everyone answer this question. Yeah, Shakya, absolutely you were right. Absolutely. No, it's not A, it's B. Absolutely, absolutely. All okay, because virus they have capsids composed of protein, but virus they don't have capsid. Viroids are just like RNA molecules without any coat, any capsid. So B is the right answer. Okay, you are right, Shakya. Absolutely right. So B is the right answer. Okay. Next question. It's a very simple one. What is the name given to virus that are single-stranded RNA that acts as a template for DNA synthesis? Okay, what is the name given to the virus that a single-stranded RNA acts as a template for DNA synthesis? So easy. Come on. Okay, so it's very simple. Definitely, I think all of you know that. It's retrovirals, right? They have that reverse transcriptase and they can make DNA from their RNA. That's what it is. See, they can make DNA, okay, DNA from their RNA molecule itself. See, from RNA to DNA they can make. So retrovirus, reverse transcriptase. I think all of you know that. Okay, so next question. Most molecular biologists think that the virus originated from fragments of cellular nucleic acids. Absolutely. So which observation supports this theory? Okay. It's a good question. Definitely. Okay. So or even in human genomes, there is a huge percentage of of our genome is have a huge similarity with virus genome. Exactly. Okay, because viruses are so primitive, right? Okay, so basically, viral genomes are usually more similar to the genome to the host cell than to the genomes of viruses that infect other cell types. Exactly. Okay, so they have a very similarity. The evolution takes place in a way because of the similarity that all those viruses, they have their own host range, because the virus that will infect the host and the host cell, they have a huge similarity in their genome. That's what it is. Okay, and that's how the host pathogen interaction or virus host interactions, okay, and these evolution have taken places and stabilized, okay, in course of time. Okay, so that's why D will be the right option. I mean, this can support this particular theory. Okay, that virus actually originated from fragments of different cellular nucleic acids from different origins actually. Okay, and that's why even in our genomes as well, our genomes are made up by almost all those different viral fragments. We have several similarities of different sequences out there that we got from different viral progenitors. Exactly, course of evolution. Okay, it's a recent report, so you can check it. Okay, that even in our genome there is a huge percentage of sequences that we got from different viruses. Exactly, so D is the right answer. That's why. Okay, next. A bacterium infected with an experimentally constructed bacteriophage composed of T2 protein code and T4 phage DNA. Okay, T2 phage protein code and T4 phage DNA. Okay, protein code from one phage, DNA from another phage. So the new phage will have what? Go for it. Okay, I think all of you can understand this one, because the new phage, okay, the DNA they are getting from T4, because whatever protein, whatever that particular phage virus will produce